This is a production of Cornell University.
So today, my talk, first another thing I want to thank Brian for his kind introduction. It has always been a great pleasure for me to work in this lab since I was undergrad. And I believe that will convey to you through my presentation. Okay, so today we're going to talk about motility regulation in this pathogenic system, Pseudomonas syringae or tomato disease 3000. And the first thing I want to touch on is to show you a little bit about this bacteria. So here you have a picture of our rod-shaped Pseudomonas cell. This is original cell, unfortunately not a syringae, but this is the highest resolution I can find on the internet. So you can see that we have a rod-shaped cell and with polar flagella, which we will touch on in a minute. But our species, Pseudomonas syringae, is a plant pathogenic bacteria that has a wide host range because there's over 60 total pathivores. The model strand we're interested in is DC3000, which is a tomato pathogen, but can also affect Arabidopsis as well as tomato. This strain has been developed as a model strain largely thanks to people in this department on this very campus, because people like Alan Comer and Greg Martin, we know a lot about their type 3 secretion system and effectors. So, but today I'm going to talk about not the virulence part of it, but the motility part of it. And I got into this because when I joined the lab back in 2017, Swingle Lab has already been working on motility for a while. And this is the result at the time when I joined the lab, where you have Pseudomonas disease 3000 use its motility to evade plant immunity, especially the part of PTI, which is where the plant are detecting flagellin. So without going too much into this the detail about this model, which concerns a chapter of my thesis that I won't be present here today, but will probably come up in my B exam, I just want to say that at the time, we already know a little bit about motility control in disease 3000 has this ecological function. So what motility are we talking about? There are total 5 different types of motility that are present in bacteria, and we're mostly concerned about swimming motility in this strain. So several features of swimming motility I'd like to point out is 1, it requires the bacteria to be inside of a certain liquid or semi-liquid environment. 2, it needs this flagellum. It needs flagellum as the main organ, if you would say, or as a main structure it relies on to facilitate the movement. And 3, it is consuming energy. The flagellum rotation relies on proton motor force to power itself, to generate the torque and pull the bacteria forward. Now, motility in bacteria in general has been topic of study for past half century or more. And the most well-known model of motility regulation in all of bacteria is chemotaxis. This is a kind of an algorithm built in the bacteria, if you say, that follows a very simple logic. It always wants to improve the chemical environment for the bacteria. And here I'm showing you that if you put a bacteria, a hypothetical scenario where you have from cold color to warm color, the concentration of say a nutrient or some kind of chemical the bacteria really likes, and you have our polar flagellum bacteria such as Pseudomonas moving or swimming inside this environment. So what you would observe in any case would be kind of a run and tumble model, which describes the movement and the sudden break and then the disassociation of all flagellum, of all the polar flagellum, and then reorientation at a random angle. And it's also described as a biased random walk because the randomness of the break, but also it's biased towards in such a way that when it's going above or when it's climbing the gradient of the nutrient or the chemical it actually likes, it has a lower probability of stop and reorient. So the biased random walk well ended up help the bacteria move to a higher concentration of the chemical that it actually likes. So Pseudomonas is a well annotated strain. It's one of the first. DC3000 is one of the first fully sequenced bacteria, plant pathogenic bacterium. Thanks to that, we can fully transfer the knowledge we know on E. coli and all other bacteria about its chemotaxis regulation into this strain. Now, this is a very complicated system, and I won't go into much detail about it, but all I want to show here is that we know what these genes are in our genome, and we can find them by any kind of like screening we have. So let's touch a little bit on what motility can do in general. The most basic function of motility in any bacteria is try to get to different location where there are nutrients present. And it's kind of like the two sides of the same coin that you can also kind of get away from your clone mates. So as a group of the same identical strands, you are evading competition with your king. And that way, your group in general or overall will have a higher efficiency at uptaking nutrients into your organism and help with reproduction and so on, or other important functions. So as I alluded to a little bit earlier, our group has been working on the bacteria using its motility to evade plant immunity. So what other functions can motility achieve in a plant pathogenic bacteria? Well, in our example, the plant DC3000 getting to plant apoplast, and the challenge it's facing there is that the PTI is detecting it. The PTI is detecting it using flagelling and so on. So that adaptation, as our lab has previously found, is that it can use LGU to downregulate motility as well as flagella synthesis there. You can apply the same logic to other locations where the plant pathogenic bacteria can land on, such as leaf surface, before they enter the apoplast. Steve Lindau's group in UC Berkeley has previously found that some of the challenges plant pathogenic bacteria can face on top of leaf surface includes it has very unevenly distributed nutrient. And as we all know, as if you're ever taken 3010 or 4010, you know that there's also very limited entrance into apoplast for any bacteria pathogen of plant. So that could potentially be mitigated by chemotaxis. There has been literature showing that bacteria can use some kind of organic acid as a signal they're tracking to find those natural openings and wounds. Well, but there are also other known challenges that are present on the plant surface, such as the extra environmental hazard from the sun, the UV lights, and also Steve Lindau's group has also shown that there's very high osmotic stress on top of that. And another thing that haven't been discussed a lot in the literature is on top of plant surface, you can have the competition with the epiphyte microbiome. And we don't yet know a whole lot about what the bacteria is doing to deal with or to cope with those challenges. But as a small spoiler, I think we have found some evidence in our screening, and later I will show you the data that they might have some mechanism already in place for that. In addition to that, I'd like to also introduce to you the idea that DC3000 is a social bacteria in that it engages in social behavior such as cell-cell communication. Here I'm showing you that it does harbor a typical quorum sensing system. This is how the bacteria is most famously known to interact with its own species as well as some other species. And they can co-coordinate expression of genes and so on. And DC3000 also engage in production of public goods such as surfactant and siderophore. So here you're seeing the typical or the most famous yellow pigment pyoviridin in our strain. So with all that, I'd like to present a question I'd like to discuss today. That is, what ecological functions can DC3000 achieve with its motility control? Because now we already kind of know it can do a little bit of dealing with the host, maybe help with its virulence and such. And with that, I'd like to present the first two chapters of my thesis. The first project I want to get into is a TN-seq-based screen of novel swimming motility genes in D63000. This is a collaborative project between our lab and the Philosophical lab, mainly with Dr. Tyler Hellman, who joined us a couple of years back as a postdoc. So at the time, we already know that we want to find novel motility regulators in D63000 because the LGU, we have done a couple screenings with the LGU project and with various success. And Tyler at the time joined us with a very powerful tool that is TN-seq. So what exactly is TN-seq? Well, TN-seq in its essentiality is asking the same questions over and over again across the genome. That is, if a strand has a point mutation, in this case introduced by transposon insertion, in a given locus in the genome, does that strand grow better or worse under any type of environmental selection you impose or not impose? So you can imagine that we have two strands here, each represented here. Those black dashes represent their relative abundance or how many copies they are in a population. The first strand we have here has a copy, has an insertion on the tip of gene A, and the second strand we're talking about here has an insertion that at the end of the gene B. So what you can do here is you can pull this library population, pull them, and let them go through some kind of selection. And after they grow, you can sample the posterior or the after selection population, and then count the relative abundance of the copies of each of those strands. And with that, you can see that gene A in this hypothetical scenario, its abundance decreased after the selection, whereas gene B remained more or less the same. And with that, we can say, oh, gene A, the loss of function in gene A is preventing the growth of that strand under our selection criteria. So with that, we can scale it up and make it a full screen. Instead of just one strand per gene, you can have multiple now. And so for each one of those strands, you can calculate the relative abundance of the before and after our selection in the respective populations. And with all of those strand fitness or the strand relative abundance, you can calculate the gene average fitness. So that will be a ratio that is either below 1, equal to 1, or above 1. And this helps us to establish a link between a growth genotype to a gene's function to a gene's contribution to a selected function. And this selection is totally arbitrary. We can manipulate it as we want. And Tyler come to us with this method, as well as a library that has been generated by Dr. Jennifer Lewis in UC Berkeley, which has been mapped but hasn't unfortunately been published. And another note here is that in the data I will present you in the following slides, those ratios will be converted by log scale. So everything that's below 1 will now be negative, and everything above 1 will be positive. Well, so now we have this powerful tool. The next thing to figure out is how do we link that to motility, because we're essentially interested in motility contributing genes. Well, at the time when I joined the lab, this method of using soft agar swimming media to quantify the swimming phenotype is already long established in the lab, and also it's a standard method in the field. So what you do here is you just inoculate your bacterial strain in the middle of a 0.3% agar plate. In this case, I'm showing you a minimal medium that is MOPS-based, and you let it grow. For D63000 in this particular media recipe, it usually takes 48 hours for you to get to the size of decent measurement. And if we're dealing with some kind of mutant that does not actually move or somehow have impaired motility, the colony simply does not expand from the inoculation point. So the logic of this assay is that the low motility strands cannot move far from the inoculation point. So how do we incorporate that and the TN-seq method to make a screen. Well, the most easiest thing to do is just like brute force and fuse them together. So, well, that's what we started doing. So we take the library culture and we make an inoculum from that, which is easy. And we take the time zero sample as you do in all TN-seq experiments. And we just inoculate and let the swimming assay go with a library. So this is not a single strand, but a pool of bacteria strands that each has a single mutation somewhere in the genome. And we let them grow. We sample everything that's in the red circle, which is a whole colony. And we use that as our samples to repeat this process, or we just, and in addition to that, we also keep a sample for DNA extraction and BARSIC. So in that, you can see that the selection we're imposing here is not much. It's just the growth in the media recipe we provide them, and as well as the swimming agar. So the expected hits we are looking for here is that we're looking for nutrient restrictions because this is a minimal medium, and also metabolism genes that help them going through those nutrients. But we're also thinking, because this is a nutrient limiting environment, so motility will help in this case. And we're not sure how many of those we will find. So let's just roll with this and set up a baseline. But in the end, what we found is we actually hit quite a lot of motility genes. So here I'm showing you the 4 tiers of flagellar motility regulation that is present in Pseudomonas aeruginosa, and I'm transplanting that here in DC3000 genome. So here you can see some of them are in white. Those are the ones that are missing homologs in our strand. The ones in gray are the ones we didn't find in our mutant library at all. And then the red ones are the ones that we found across the 7 replicates we did with 7 total passages we did. If at any point of time, they have a significant penalty for loss of function. So that means those strands, if the bacteria has a loss of function in any of those genes, they're not growing, or we're not getting them from the final samples. Black means black means exactly opposite of that, meaning that they are always being recovered. So we found total 34 out of 47 of this. And that is pretty nice, but also we're still facing the problem that we need to distinguish them from the nutrient restriction genes and metabolism genes. So to counter that, after a long time of trying to figure out what to do, I came up with this scheme, which turns out to be, I think it's intuitive at least, is that if we add another layer of selection, what if now I select against motility? If the environment itself is selecting for motility, and I then add an arbitrary selection against motility, doesn't that mean only the motility genes will respond to this selection? They are still growing in the same nutrient condition, and also they should, though if those genes are having high phenotypes, they should be present in both groups. But if there is a difference between those 2 selection schemes, those should be the response I detect from just the motility contributing genes. So we went with this model. And the first thing we expect is, well, now we're selecting against motility. So the motility should decrease on population level. And voila, they did, which is pretty neat. And when we then look into how the finis for genes are behaving across these passages, we found a very clear split that when we're selecting for everything, the population kind of evolved this way, and when we're selecting just for the non-motile population in the middle of the plate, the population evolved in a very different trajectory. So what do we find? So this is a good start. We think we're hitting something. So we look into the highest finis values or finis phenotypes we have in all of these lists of genes, and we found 3 groups of them. that are behaving very differently. The first group, represented by the chemotaxis genes, Kb and Ky, these are known chemotaxis motility contributors. They have a fitness that is lower when the whole colony is passage, when the plate itself is selecting for motility. But when we then added the extra layer of selection, they reverse back their fitness to be positive, meaning now losing those genes are not as bad for the strands once we select for low motility. The second group, represented by flagellar motor genes, MOT A and MOT B, do the exact opposite. Their response is flipped. And that is still a mystery to us why they will do this. And then the third group of genes, which I think most of this audience will be interested in, is why we hate type 3 secretion genes. They are active and they are having a positive fitness scores in either cases, meaning that they're on in this minimal medium situation. But MOB's minimal medium is not the usual hard minimal medium pH. So we're very curious why they're actually on here. And we tested them, they didn't really do anything to motility, unfortunately. And those three groups, when we plot out every gene and their responses across all our passages, you can see a very clear split of these three groups. So because we have so many more questions about the second and the third group of genes, we ended up focusing on the chemotaxis genes because we know what their phenotypes should look like once we go into individual mutational study of these genes. And the three members, other members of this group are PSPDO406, which is a cyclic GMP enzyme. So this kind of like links it to motility in its nature just by what we know about cyclic GMP. And we find that, well, indeed, you have a phenotype where you knock it out and it's slightly, it impairs motility. And when you complement it back, the motility come back a little bit, not fully, but partially. Also, this is an overexpression instead of its natural promoters. 1042 is another interesting one because this is an anti-sigma factor for oxidative stress that has been shown by the previous member of Philosophical Lab, and it has not really been linked to motility or confirmed to link to motility in the past. And we found it has a very clean impaired motility by knockout, and overexpression completely restored the motility phenotype here. So that's telling us that there are linkage between stress response and motility control. The ecology of it, we don't know yet. And then the oddball of them all, the hypothetical protein PSPTO4229. Absolutely stunning result in that knocking it out does not really do that much. It slightly impairs motility, but when we overexpress it, it abolishes motility. So very odd, and we don't know anything about it. So that's where we get sidetracked into go full in on 4229. So this is a hypothetical protein. As many of them are, they're small proteins. It has a total amino acid 273. It has a only annotation on the protein is a HDOD domain from 23 to 210, which does not tell you really anything. HDOD domain is such a general term that incorporates all kinds of functions. It doesn't really contain much information. And I'm showing you kind of a model here. So we tried a whole bunch of methods trying to figure out what this protein is doing. The one that ended up working for us is error-prone PCR. So this is we generate another library. Well, that's a lot of library generation within one talk. There's one more. So we generate a library of sequence, each with few single point mutations by error-prone PCR. So this is you're trying to use a different polymerase and so on, trying to get error sequence happening during PCR. And then we fuse that, we put all of them into our expression constructs and ask them to perform on swimming media. So what happens there is, because we know the wild type 4229 overexpression phenotype is complete abolishment of motility, it's very easy to screen for them. And we ended up getting 40 different plasmids that are having failed to suppress motility phenotypes. So here I'm showing all the sequence we got, amino acid sequence we got that are still full length. And I'd like to diverge for concentration into the conservation plot here in yellow. these are where we found among those full-length sequences that are having mutations from the wild type, which is on top. And also we use this tool called ScanNet, which can predict protein-protein interaction by just inputting amino acid sequence that is recently developed. And that gives us a prediction on each amino acid residual, how likely they are involved in protein-protein interaction. And we indeed find the highest predicted interacting site is kind of like aligning with a hotspot here. So we went in and we did the mutation. We replaced those two arginines that are being flagged out with alanines. And we found that the phenotype is gone. So great, now we know there is an important residual for this suppression phenotype. The ecology of it, again, no idea. So what I've told you so far for just one of those, for the first project is that one, TN-seq is, again, very powerful, as Laurie told you a couple of weeks ago. And as long as you can select a function, this is a tool that can get you most of the genes you're interested in or give you great candidates. Second, swimming motility in disease development can be co-regulated by stress response with no previously known connections to motility. 4229 has a dominant suppression phenotype on motility. It's swimming, but as an additional note, it's also swarming. I didn't show the phenotype here. And yeah, because we know nothing about the ecological significance of this, there's a lot more study to do if we want to follow up on them. So I hope this has piqued some of your interest in motility because the next story is going to be a little bit more wild than this. I'm showing, I don't know how much it's showing here, but I'm showing you a very weirdly shaped disease-resistant colony here. And this is something we found almost coincidentally or by accident back in the days. And this is a socially induced disease-resistant swimming motility we call baited expansion. So back when I joined the lab, Brian and Kern was interested in the phenotype when you put two colonies of the same species, bacteria, onto swimming media, they somehow form this gap and they behave slightly different than they were if they're growing alone. So I had the job to replicate this phenotype a bunch of times. And in doing that, we also incorporate the interspecies interactions as sort of like a second control for these phenotypes. So in doing that, what I realized is Pseudomonas is doing something weird. When Pseudomonas is paired against Dickia on third day, or paired with E. coli or Xanthomonas on fifth day, you start to see the colonies are doing something on these edges that are closer to the interaction partner. And I was very interested by this. I don't know what they're doing, but I want to study it. So I tried to standardize it. The problem is these colonies are not really standardized. This one is on day 3, this one on day 5, day 5. So I need a way to bring them to a more comparable level. So my solution, because the strands we're using are model strands and are strands with more genetic resource with them, I was able to get non-motal mutants of them and standardize this phenotype to such. So you can see that when you have an unbaited control of disease result, then it's more regular, it's radial and symmetric. Whereas when you have an E. coli bait here, it starts to behave non-symmetric. The same with Dickia. But when you put another disease result and as its interacting partner here, it's back to being radial. So great, now we have a standardized phenotype. I can start to quantify things. The first thing we quantify is how much growth they are having on each side, or how much the bacteria colony is expanding on each direction regarding the position of the bait. So here you can see the blue lines here represents the proximal because it's proximal to the bait, and the red is the distal because it's away from the bait. From the both starts at the inoculation point. And the quantification of that is laying up here. You can see that with E. coli or Dickia as a bait, the overall colony expansion is up, but within the two sides of the colony, the proximal especially is induced by the presence of the bait. And we recently come up with this actually, is to trace the colony edges and then just overlay them to show that there is a significant difference in how the colony morphology is happening. So here you can see that even with a control plasmid, so they're growing slightly slower than the previously shown, you still have the extra growth or expansion on the side that is close to the bait, proximal to the bait. So the next question I had for this was, is this chemotaxis? Because it looks very much like chemotaxis to me for two reasons. One is directional. Chemotaxis is always directional. And two, we observed in many of our replicates that if the closer location of a single bacteria is located, or of a colony, disease-resultant colony, to a bait, you seem to get stronger effect. So those are chemotaxis-like features of what we're seeing here. However, our collection of chemotaxis mutants are usually non-motile, which means you just don't get motility and you can't really use them to test whether they are behind this phenotype. So the next thing we could do was to test for inducibility. So the idea of that test is if you have a chemotaxis-like behavior where it's tracking some kind of cumulable chemical signals or depletion of that, you can just let the depletion or accumulation happen for longer, and then that should provoke even stronger response. And we kind of find that. This is a pylon I'm showing with Arvernia amylovora EA273 as a bait. And you can see that this bait here has been inoculated 24 hours before the disease-resultant colony is inoculated. On the right, you have a bait that's the same species of Arvernia that's inoculated at the same time point as your disease-resultant. And you can see a very clear behavior difference. This one has stronger behavior. We can also replicate that with the tracing, but with E. coli, it still provokes a very different colony morphology, but in this case, it's more lateral expansion. So it's more on the sides of the colony. So with that, our conclusion here is that it is something that's accumulating. It's either accumulating of a signal or depletion of a signal, but it's accumulable in its nature. The next thing we did here is we're interested in what is the driving force for this behavior to manifest. And at the time, we have two hypotheses. It can be either growth difference, so maybe this part is growing more, or it is a major motility difference. And to test that, what I ended up doing is I ended up cutting the plate in half. So that separates the plate, whole plate, into two sides. One is proximal to the bait, the other is distal to the bait. And I'm curious whether those two parts of the colony are actually having similar amount of viable cells. So we did the test. And indeed, when a colony is baited, the proximal side is having more viable cells than the distal side. Whereas when it's just a normal colony growing on its own, that didn't happen. But to our biggest surprise here is that we couldn't detect the immediate finished benefit of doing the behavior because the total viable cells in the colony is not significantly different. So that pushes us more leaning towards this is a motility phenotype rather than a growth phenotype. So we recently, with the help of the new fancy microscope we got, we were able to look at that more closely under that. And here I'm showing you on the top. So in this panel, the bait is towards the left. On the top, I'm showing you the sampling, a field of view that's fixed coordinate on the proximal edge of the population, and on the bottom is on the distal. And you can see that on the proximal, you have, so the disease-resultant here is tagged with GFP. And you can see on the proximal edge, you have faster growth. The intensity changes over the 90 minutes course a lot, whereas on the bottom, there's not much. But also, you can see the movement of the cells. So it might be a little bit hard to see on this resolution, but the first white line is where I marked the initial edge at the beginning of 48 hours post-inoculation is. And this line here is where I could, on a higher resolution, find the edge of this colony. The same is for the bottom. So the short of the story is over 90 minutes of observation, the proximal edge moved about 600 microns, whereas the bottom edge, the distal edge moved only like less than 200 microns. So we're seeing that there is a movement difference here. Well, since we're seeing that much difference in behavior of these two edges, we're now interested in if we can find any genetic markers for them. So the approach we took here is we want to sample those edge populations and see if we can find a transcriptomic difference. And indeed we do. So here I'm showing you exactly where we sampled on each plate. We used the most distal edge or the most proximal edge to abate within our DC3000 colony, and we run RNA-seq with those. And at the end of the day, what we found is the proximal edges that are most close to a E. coli bait and the proximal edge that's most close to a DK bait are behaving very, very differently than all the other edges we were able to sample, and those edges kind of mix together, which is more clear on this PCA plot. So you have the Dickia in blue and E. coli in this red eclipse. So the biggest, so we went into the list. We think we found something, and the 1, 2 is a list of differentially regulated genes. The biggest fold change we found was with this hypothetical gene, another hypothetical gene, PSPTO 5639. Does not have any information, but in the very close neighborhood, we also find this MOA gene. And we were able to confirm both of them are indeed induced in that in the proximal population towards the bait by QRT-PCR. So what does that do? MOA is annotated, and we know from its annotation that it encodes molybdenum cofactor biosynthesis protein A. It's also a GTPase for what we know. MoCo is a highly conserved function across bacteria species and also across kingdoms. So even humans and plants have molybdenum cofactors. They're involved in redox enzymes, and also those are involved in global sulfur, nitrogen, and carbon cycles. MOA, again, additionally, MOA was the only hit among all the RNA-seq data we have within this MoCo function, within all the genes that are linked to molybdenum cofactor function. So, well, but we're geneticists, what can we do? We knock it out and find out that it somehow mitigates or eliminates the phenotype. But when you put it back, you have the extra growth, as you can see, the tiny like wild type control here. When you add it back by overexpression, that extra growth come back. So great, MOA is our guy. But is it the GTPase part or is it the MoCo part? Because GTPase interact with GTP that also can potentially interact with cyclic diGMP. So we're really curious which approach did it take to actually influence motility and in this case, our behavior. So we look into what's in the MoCo biosynthesis, and this is a MoCo biosynthesis genes in E. coli, and we picked another essential gene for this biosynthesis function. We picked the MOA, which is the last in the step of MoCo assembly, where it inserts the molybdenum actually to the cofactor complex. And we did the genetic thing again by knocking it out. And again, we were able to see this phenotype or this behavior disappeared when we knock it out and come back when we overexpress it. So great. Now we know MoCo is our guy instead of just MoA. So what I have told you so far is that in this chapter is that swimming motility is socially inducible, and the behavior we call beta expansion is tracking some kinds of accumulable signals, be it the actual accumulation or depletion. We also know that beta expansion results more from motility rather than growth from our extraction or viable cell counting assay. And last but not the least, MoCo contributes to the development, and it seems to be essential to the whole behavior. And now we enter the wild speculation part. Is it just me, or does this look like a charge to you too? Like this looks very aggressive to me, right? So we don't yet know that much about its ecology. We have speculations here. And one of our speculations is that this is a competition behavior and it's trying to get closer for either territorial range or get into a closer position to initiate one of the cell-cell interaction mechanisms such as type 6 secretion or actual cell killing. We have no information of those genes being upregulated in our RNA-seq data at all, not type 6, not any like toxin production or anything like that. But we cannot rule out once they get there, they might do it. But again, this is just fascinating. And I've been looking at this and think of this for last 6 years, and we still haven't cracked it yet. So as a summary, I hope those materials have piqued your interest in motility in our very closely studied DC3000 bacteria. I hope that helps you appreciate that motility regulations can respond to social interactions as well as environmental stress. And there's a lot more ecology to be studied in this system. With that, I'd like to thank the past and current Steve Swingle lab members, our collaborators, Dr. Jen Lewis, Dr. Tyler Hellman, Paul is in the audience, and the Cornell T-Rex and BRC that helps us with a lot of the sequencing stuff we do. I'd like to also thank my committee members and everyone that I interact in the office daily, as well as the amazing postdoc and graduate student body. I'd like to make one more comment. I have a bunch of friends who told me I'm the happiest graduate student they ever seen. And I think that's largely because I'm in this amazing community and I work in an amazing lab. And with that, I'd like to have my birds take questions from me, but I will speak for them.
So any questions for Igor?
Josh.
So in the second part of the talk, bacterial baiting and such, the ones that it's swarming towards or charging, as you put it at the end, can it eat those bacteria? And or if you mix them together or co-inoculate at the same time or put them really close to each other, does the Pseudomonas kill the E. coli or whatever else is there?
So we have done some assays. Okay, so the question is, if we just co-inoculate the two bacteria together, are there evidence of killing happening? We haven't been able to observe any killing with stains and stuff yet. We were able to see suppression, but if we co-inoculate in liquid culture, but that could also just be nutrient competition instead of active killing. We don't have a lot of evidence on, or we have very few evidence on that actual toxin production or anything is involved in these interactions. So our main hypothesis right now is that it's a competition behavior, but it's on the level of exploitative competition where it's just trying to grab more nutrient for itself. Lori?
Really great talk. I had a question about your TNC data. So you pulled out these type 3 secretion signals. Do you think, since this is I guess a social interaction, do you think there's a possibility of another way that you could look for things that might be signals that are going between cells? Presumably those would not come out of a TNC because they'd be shared amongst the population. But is there some other way you could look for what those type 3 secretion signals are like what they're signaling or what they're sensing or whatever?
Yeah, so the question is there alternative methods to screen for or look for like a signal that is being transferred in between the bait and the disease resistant colony? Our lab has done something before trying to go like, mass spec and stuff like that. We didn't really get anywhere with that. I remember so that happened when I was still undergrad, I think. And another undergrad at the time, Mikhail, who was working on this. And I remember the comment he made was there's so much processing so what he was trying to do was trying to extract the gel in between the colonies and trying to figure out if we can find something in there. I remember the comment back then was, though they were super processed, like there are so many layers of processing that he was pretty negative about like potentially finding anything. And I don't think we ended up finding anything. Okay.
I was gonna say, like, do the secretion genes you found, is there anything known about what they're transporting? Like, did that help get at that at all?
The secretion genes I found, are there any information about what they could be? So for the social motility, so MOA-A is not a secret. So molybdenum cofactor in general are not, well, I think all of them are not secreted, they're cytoplasmic. So that actually didn't point out to any directions that leads to like a potential secreted signal into the space.
So I guess I was talking about the hits from your TNC screen, your type 3 secretion hits.
Oh, okay. So that actually does not relate to the motility, so social motility. Yeah. Adam.
Follow-up question on the type 2 secretion genes you got in that hit. You said you were surprised by those hits because those genes aren't even expressed under those conditions. Did you confirm that they're not expressed?
Okay, so Adam's question is, did we confirm the type 3 secretion genes were active in the MOPS minimal media condition? We did not go into that route. So when we found that the type 3 secretion genes are there, what we did is we pulled out all the mutants we have on type 3 and did motility assays with them. And we found that they weren't really affecting motility. So the only other explanation is they were on and they were taking a toll on the metabolistic or on the energy for the bacteria so that their growth might be negatively affected by having them there. That's our hypothesis for why we might have those hits. Zoom chat. A lot of congratulations. Thank you all. But no question.
So no further questions. Let's thank Igor.
This has been a production of Cornell University, on the web at cornell.edu.